Hello, today I'm going to be talking to you about the Protein Data Bank, which is a website that we use to get our files for our docking programs, and also it's a resource you can use if you want to know anything about sort of biology or biochemistry or proteins in general. So before I start, inside the folder with this video, there's also this PowerPoint, which has sort of pictures of everything that I'm going to go over that you can use to follow along and as a refresher if you need it. I'll come back to this PowerPoint at the end for these last two slides, but just know that I'm not going to be using these as a description. I'll be doing it on the actual website, but you can still use this to follow along or as a refresher as needed. So when you first get to the Protein Data Bank, which you can find by typing in rcsb.org or by simply searching in Google Protein Data Bank, and it'll be the first result. One of the things that you can really kind of start looking through is you can find any information on it, really any enzyme in here. But of course, we're interested in aromatase. So to get to there, we're going to come up into the search bar here and type in aromatase. Now, you don't want to click on any of the things underneath of it here, because that is going to give you very specific results. It's not going to give you everything that has to do with aromatase. So it's best just to leave this and just hit enter. And then it's going to give you sort of every structure that is based on aromatase. So what all of these are, the reason that they're all different, these are all different versions of aromatase that people have found in these are all different papers, right? And people have found these structures by crystallizing aromatase and then using usually X-ray diffraction to determine what the structure of the protein is, right? So each one of these is usually crystallized with a very specific ligand, right? So this one is androstenedione, right? That's the ligand that is in complex with. But as you go through, you can see a bunch of different ones, right? And you can also see that there aren't all the same version of aromatase, right? So one thing that's really important to note, this word right here, CYP19A1, that is specifically the type of aromatase that we are looking for in our group, and that's what we are specifically trying to get. So if you're going through here and you're trying to find a different type, a different folder or type of aromatase to use, try and make sure it has that code in it. If it doesn't, you should definitely double check before you use that that structure, because it may not be the right type of aromatase, right? Aromatase exists in lots of different forms, right? And we want the CYP19A1 form. Additionally, another thing that's important to note is all of these methods here that you can see have a this sort of 2.9 angstrom or this value next to them. What this value means is that that is the resolution that the X-ray diffraction was held at when they found this structure. We want the lowest possible value because you might not have been through sort of an instrumental class yet, but resolution, what it means in instrumentation is it is the smallest distance two data points have to be from each other, and that can still be distinct. So this means, this 2.9 angstroms right here, that this X-ray diffractor, when it was measuring this, could take a measurement every 2.9 angstroms, and those two measurements won't ever overlap, right? They won't like blend together. And so you want that to be as low as possible. In general, we want our resolution value to be less than 3, because when there's very good new technology out, and so having things that are less than 3 is very good, and that's usually better. And then finally, the last thing that you kind of want to think about if you're perusing this and trying to find your own file is all of these are papers, so they have a publishing date, right? This one right here at the top, 3EQM, that's the specific code for this very specific structure and paper, was published in 2009, right? And so what we want to do is if you're looking for a new one, you want to try and make it as sort of modern as possible, right? Like this 1999 version, we don't want to go back in time, right? Because, you know, what's the point? We want new data, right? So, for instance, a good example way down here is this 5JKV. This was published in 2018, and it has an X-ray diffraction resolution of 2.75. And if you read through the title, you can see it is CYP19A1. So this would be potentially a good one to use alternatively if you were playing around with that or you were asked to find a different one, right? What a good thing to mention is that this does not use um, androstenedione. This is using polyethylene glycol, which is a different ligand. So you got to be careful if you're using something different, right? Always check with Dr. Ma before you use anything different. 3EQM is the one that we normally use. So when you pull up this page, you can see all of this information right here in the search bar. But if you click on the actual name right there, it will take you to the actual page for this structure. And this has a lot more information on it. I'm not going to go through all of it. You can kind of go through it on your own and see what it is. I'm just going to go through the most important parts. One thing that's very, very important here is that you can find the literature right here in this page. This has all of the literature on it. And another thing you can do is if you, at the top here, there's a bunch of tabs. If you click 3D View tab, it's going to take a second to load because it's going to load the 3D viewer. But what this does is this generates a 3D structure that you can use to look at. So if you left click and drag, you can rotate the molecule around, right? And you can see way down here in the center, it's hard to see. If you scroll in with your mouse wheel, you can zoom in and you can zoom back out in the same way. It's a little hard to see, right? There's a whole bunch of stuff going on, but you can see sort of this group that I have highlighted right now. If you hover your mouse over any group, it'll highlight it. This is the heme group, right? So this has our iron and everything in it. And right here is our ligand, right? Right there. That's our ligand. And if you click, it'll zoom in, and you can get a little bit of a better look at it. And this ligand is what we end up actually replacing, and we'll talk a little bit more about that at the end here. But this is how you know it'll be different. If you get a different ligand, so if you got that polyethylene glycol one, this molecule would be different, right? So you can click again to unzoom, right? You just click on the white space. Another thing you can do is up here in this sort of toggle or in this settings panel right here, you can turn on spin, and you can even adjust how fast it spins, right? And this is really good for if you wanted to create a GIF using the screen to GIF video and software and put this in a presentation. This is really nice and appealing to the eye, and it definitely shows people that you have the 3D structure and you understand it. You can also, if you're doing this, you can make this full screen by clicking this button right here, or expanded view at least, and then you can get an even larger picture of it, right? So those are some really cool, and it has a lot more features in it that you can do. You can change the background and stuff. And so this would be really nice for getting that. Don't forget that in the computer modeling software that we use, we can do a lot of these functions already as well. So don't think you have to use this one. It's just another option for you, right? So let's close this expanded view. And there's some more things that we can do with this. So if we come back here to structure summary, right? So this is that summary page. Right here on the left, you have the picture of this, right? We clicked on 3D view to get to that viewer. You could also click on structure, and it would take you to that exact same page, right? We don't really worry about electron density. That's not really what we care about. But ligand interaction is another interesting 3D viewer that you could look at. So if you click on that word, ligand interaction, right? It still pulls you over to the 3D view, but it shows you all of the ligand interactions that are happening inside of your molecule, right? So you can see here, there's this, oops, let's zoom back in here. There we go. You can see that there is different ligands, and you can change what all of these mean, right? So there's your ligand and waters and all of this good stuff. And this gives you a pretty good idea of all of the bonds that are going on, right? The hydrogen bonding, any sort of dipole-dipole moments, or anything like that that it might be going, or chelations. And this is just really interesting to look at. But do be aware that you can see this much easier in our computer modeling software. So generally, you don't want to use this as your picture-taking method to take a picture of all of those interactions, because we have a better way to do it later. But it is just kind of cool to look at, and you can kind of see everything that's going on inside the molecule, right? So there's one last thing that we need to go over back over in the structure summary. If you want to actually use this structure in the computer modeling, we need to download a very specific file that we need to use. To do that, you want to come over here to this download files button, and when you click it, it'll open a menu. The format that we always want to save it as is this. It's called PDB format, right? When you click that, it'll download the file, right? And if you want to use it, you need to move it into in our research folder in the docking and in the aromatase docking, right? Here is 3EQM.pdb, right? That's this molecule in the background here that has been saved as this file. But again, you want to double check with Dr. Ma before you do any of this, because we don't want to start putting random files in here without approval, because we don't want to mess up any of the docking or anything like that, right? But if you did have a structure that you thought you wanted to use for some reason, that is how you would save it. Is again, you come to download files, this blue button up here in the top right corner, click it, and you click PDB format, and it'll just automatically start downloading as that format, right? So that's all for the website. Let's kind of come back to this PowerPoint here, and we're going to go through some of these last two slides a little bit, just a little bit of the purpose behind sort of why we are looking at these and why we care about these 3D structures, right? So you might not be familiar with our computer modeling software, but we have a computer modeling software, that lets us put various molecules that we can design inside of that aromatase structure, and it gives us a measure of how well it binds and how well it fits inside that pocket, right? It gives us a score that we can use to kind of tell, okay, this might be a good drug molecule, this might not be, and it sort of helps us pick what molecules we really want to make and see if they're easy to make and if they actually work, and what things we think maybe this isn't going to work that well because it doesn't have that great of a score, right? So you can see right here on the left, this is a picture just like I showed you, but with a little bit more detail, right? It has everything labeled. This is showing you that binding pocket, that ligand I pointed out, that is androstenedione, right here in this sort of wire mesh frame here. And what happens with our computer modeling software is we say that that is the ligand, and that is what we end up replacing with whatever structure we want, right? And so it sort of uses this as a basis, and that's why we want to be careful if we download any other versions of that software, to make sure that we know what the ligand is, so we know exactly what to tell the computer modeling program to take out of that structure and replace with our own molecules, right? And the modeling system that we have, you can see these pictures look much different. That's because these are taken off of that software. It can do a lot of more interesting things, like it can tell you about the surfaces, right? It can tell you what parts of that pocket and molecule are polar, or what parts are hydrophobic, and what interactions are going on. And again, this is just kind of reinforcing the fact that we really care about the CYP19A1 structure. That's the specific type of aromatase that we care about, the organization, everything. And again, this is androstenedione right here as well that you can see. And you can see this is sort of highlighting a lot of those interactions. There's a lot of hydrophobic interactions, right? All these blue ones. There's the heme chelation right down here, right? That heme group I pointed out earlier. It sort of has that weird ball in the middle of that strange polygon. And then there's some hydrogen bonding that happens, right? And so what we do when we substitute out our products that it makes is it erases all of this stuff, and then it calculates based on it like rotates our molecule in that pocket, finds what the optimal sort of orientation of that molecule is, and then it tells you based on that orientation what is the most likely um, sort of bonding and bond types that it will form. What's the most likely type of interactions it has? And what's great about our software, as you'll learn, is that it also tells you the distances and the lengths of those bonds, because that tells you a lot about their strength as well. That's really it for the using the protein data bank software and to get those files. If you want to learn more about the actual sort of software and how to use it, there are more videos after this one. But I hope this was helpful. Thank you very much.